uncovering hidden clues inside our DNA, how 3D genome mapping is opening new frontiers in drug discovery. Imagine if scientists could see the very blueprint of life, not just in flat lines, but as a dynamic, three-dimensional structure. This vision is rapidly becoming reality, and it's revolutionizing how we understand disease mechanisms, potentially leading to groundbreaking treatments. But here's where it gets controversial. Traditional genetic studies, which focus mainly on linear DNA sequences, often miss the full story. This is why innovations like 3D genome mapping are capturing the scientific community's attention, promising to unlock previously hidden drug targets. Advancements in genomics have already transformed many aspects of disease research. Yet, for complex and widespread conditions such as inflammatory bowel disease, multiple sclerosis, and other immune-related illnesses, effective treatments remain elusive. These challenging disorders continue to puzzle researchers as they attempt to decipher which genes truly influence disease risk without getting lost in a labyrinth of genetic signals. Leading this charge is Dr. Dan Turner, Chief Scientific Officer at Enhanced Genomics, https://enhancedgenomics.com. With over 20 years dedicated to genetics, molecular biology, and genomics research, Dr. Turner's mission is to refine how scientists identify viable drug targets. He oversees the company's scientific strategies, ensuring research efforts are both rigorous and goal-oriented, spanning everything from high-level planning to detailed experiments. Moving past mere association of genes with diseases, Turner emphasizes understanding biological causality. My role is to ensure our research is scientifically sound and aligned across the company, he shares. We focus on using the latest techniques to directly uncover how genes actually cause disease, rather than just correlating them. At the core of their approach is 3D multi-omics, a cutting-edge technique that layers information about the physical arrangement of the genome with other molecular data. This approach maps how genes are turned on and off in their natural three-dimensional environment inside the cell nucleus. It captures how many long-range regulatory elements, small regions of DNA that control gene activity, come into close proximity by folding in on themselves. Why does this matter? Because most genetic variants linked to disease are located in non-coding regions, parts of DNA that don't produce proteins but regulate gene expression. Traditional methods often struggle to interpret these variants, especially since they can influence genes from far away in the genome's 3D space. Think of the genome as a complex origami structure, explains Turner. The way it folds brings together parts of DNA that are far apart in sequence but close in space, enabling us to understand which regulatory regions control which genes. Enhanced Genomics has developed innovative assays to profile this 3D genome architecture across the entire genome in just one experiment. By combining that data with other layers, like chromatin accessibility and gene expression, they can precisely map the regulatory networks that underlie diseases. This holistic view, that's what they call 3D multi-omics, enables scientists to identify not just associated genes, but causal ones, in specific cell types and conditions. This level of detail is crucial because assuming a disease-related genetic variant affects the nearest gene often proves inaccurate. In fact, approximately half the time, this simple assumption fails, leading researchers astray. Without considering the genome's 3D structure, we risk missing our best therapeutic targets or pursuing the wrong candidates, which can be costly and time-consuming, Turner notes. Our approach allows us to prioritize targets backed by solid genetic validation, linking variants directly to the genes they influence through physical interactions. A key part of their strategy is building comprehensive maps of healthy cell types. These atlases serve as essential references against which disease states are compared. By overlaying disease-linked variants onto these baseline maps, researchers can spot disruptions in normal gene regulation, pointing directly to the pathways involved in illness. The goal? To make drug discovery more based on robust data. Turner explains, we start with large-scale genomics data from GWS studies, then systematically analyze all relevant molecular information across cell types. Because these datasets are already generated, our process is fast and thorough. From this, a long list of promising gene targets emerges, which is then refined by considering practical factors like safety, feasibility, and patentability. Rather than generating countless hypotheses and testing them blindly, this integrated, data-driven approach ensures each hypothesis is grounded in stronger evidence, saving time, money, and effort in the long run. Choosing which diseases to focus on is a strategic process. Turner mentions a two-phase method that combines historical success data with insights into difficult-to-treat diseases with unexplained genetic risk factors, especially those involving regions of DNA outside known genes. This helps prioritize areas where new discoveries could have a significant impact. Their first major focus? Inflammatory bowel disease, a condition with high unmet medical needs and a clear genetic component, making it an ideal candidate for this innovative approach. But the broader implication is that their method could revolutionize drug discovery across many complex diseases, including neurodegenerative and metabolic disorders. Turner envisions 3D multi-omics as a transformative evolution, integrating various layers of genomic data into a unified picture that reflects actual cellular biology. This comprehensive perspective makes it possible to identify causal mechanisms directly and scale discoveries across the genome. As he puts it, this technique will reshape drug discovery in the coming decade, maybe as profoundly as how sequencing changed genetics. Rather than invest heavily in uncertain hypotheses, scientists will increasingly rely on genetic validation powered by 3D structure insights, leading to more targeted and effective therapies. It shifts the focus from merely cataloging individual genes to understanding the complex networks that regulate them. The importance of this cannot be overstated. Many common diseases, autoimmune, neurodegenerative, and metabolic, have resisted traditional research strategies, leaving patients without effective treatments. But with 3D multi-omics providing a new window into disease biology, we're entering an era poised to deliver genuinely impactful therapies. As Dr. Turner proclaims, this isn't just a small step forward. It's the dawn of a new era in pharmaceutical science, where clearer insights lead to more definitive solutions, ultimately saving lives. So, what do you think? Could this innovative approach truly unlock the cures we've been searching for, or are there risks and limitations we overlook? Share your thoughts below.